This is an overview of an article entitled A-band Titin Truncation in Zebrafish Causes Dilated Cardiomyopathy and Hemodynamic Stress Intolerance, published in the August 2018 issue of Circulation, Genomic and Precision Medicine. Truncating mutations in the TTN gene, or TTntv B, are present in 15 to 20% of cases of dilated cardiomyopathy, or DCM, but also occur in up to 3% of the general population. Because of this variable expressivity, the prognostic significance of finding a TTntv in any single individual is uncertain. This is an important unresolved clinical issue that has relevance not only to patients with DCM, but also to the escalating numbers of individuals undergoing personal genome sequencing in whom TTntv are discovered as incidental findings. It has been proposed that not all TTntv are equal and that pathogenicity differs according to variant location, with DCM-associated TTntv enriched in exons that are constitutively expressed across all cardiac titin isoforms, especially in the titin A band and distal I band regions. However, these stratification criteria do not adequately segregate subjects with and without a DCM phenotype, and many apparently healthy individuals carry TTntv that would be predicted to be deleterious. This raises the key question of whether TTntv are primary causes of DCM or modifiers of disease susceptibility. The authors of the paper under discussion hypothesized that TTntv can be a primary cause of DCM. They performed clinical and genetic studies in two unrelated kindreds with DCM in which the proband was found to carry the same A-band TTntv. This prompted the authors to generate zebrafish with homologous A-band TTntv knocked into the fish ortholog of the gene, TTNA. They studied fish homozygous or heterozygous for this variant and assessed for cardiac phenotypes concordant with DCM. Here are the two pedigrees for the unrelated kindreds with DCM in which the proband was found to carry the same TTntv. The mutation is located in the indicated position in the A band. The mutation was present in all of the affected family members, as well as a few unaffected members. Heart function was evaluated in embryonic zebrafish using video microscopy. Homozygous TTntv embryos showed a severe reduction in cardiac contractility and absent circulation, as shown in panel A. There was widespread edema, and the heart chambers were virtually akinetic, as indicated in the top two graphs of panel B. The bottom graph documents that the homozygous fish were dead by the 10th day post-fertilization. Heterozygous fish survived to adulthood. However, female mutants were smaller than wild-type siblings, as shown in panel A. There were significant effects of age and genotype on end-diastolic ventricular size, with progressive dilation, as documented in panel B. The most significant determinant of ventricular systolic function was genotype, with reduced ejection fraction in both male and female heterozygous fish from 6 months of age, and reductions in global longitudinal strain from 9 months, as shown in panels C and D. Cardiac histopathology was evaluated in 6-month-old male fish. There was mild ventricular enlargement in heterozygous fish, though there was no evidence of myocardial fibrosis, as shown in panels A and B. There was preservation of a regular myofibril striation pattern in heterozygous fish, as shown in panel C. Although myofibril alignment and sarcomeric organization appeared normal on electron micrographs, there were significant reductions of sarcomere content in ventricles and atria in heterozygous fish hearts, as shown in panel D. Stress conditions can be effectively modeled in zebrafish using phenylhydrazine, or PHZ, treatment that induces hemolytic anemia in a hyperdynamic state. Heart sizes were larger in PHZ-treated fish when compared to vehicle-treated fish for both wild-type and heterozygous genotypes, as shown in panel A. There were baseline differences in end diastolic volume between the two genotypes, in each case augmenting with PHZ treatment, as shown in panel B. Similarly, the baseline ejection fraction was lower in heterozygous fish. However, whereas the ejection fraction was increased with PHZ treatment in wild-type fish, this was not seen in heterozygous fish, as shown in panels C and D. In light of these data, along with data not shown here, the authors concluded that the heterozygous fish hearts had impaired contractile responses and reduced diastolic filling with hemodynamic stress induced by PHZ. In conclusion, heterozygosity for A-band Titin truncation was sufficient to cause DCM in adult zebrafish. Abnormalities of systolic and diastolic reserve in Titin truncated fish reduce stress tolerance and may contribute to a substrate for atrial arrhythmogenesis. And these data suggest that hemodynamic stress may be an important modifiable risk factor in human TTntv related DCM.